this video, I am going to combine the Urease test and the litmus milk together. They are almost always done simultaneously, so instead of making two separate videos, I'll go ahead and make one. We'll start with the Urease, or the urea test. So you may have heard of urea before. I'm sure we've all heard of urine, but of course bacteria don't produce urine. But urea is a byproduct of protein metabolism. So as the body breaks down amino acids, it produces this stuff called urea. And if you've ever changed a baby's diaper or even smelled what smells like ammonia, that's because that in the urine you have urea, which is a protein. And if bacteria have urease, it's the enzyme that breaks down urea. The urea, I'm sorry, is not a protein, it's a breakdown of protein. And if the bacteria have the enzyme urease, they break it down and they use it for nutrients. But the product of that is ammonia. And ammonia is a very basic compound. So it's going to change the indicator to a basic color. Now with the urea, you're going to have tubes that are kind of an orange in color or a peachy color when they start out. Now this particular tube that I'm holding in my hand is a solid deep. You probably will have them as a slant, but because this is what I could find when I made the video, it's what I'm using. Just as with the previous lab, we have 3 bacteria, possibly 4. So if you have 3 bacteria, you'll have 3 tubes. If you have 4 bacteria, you'd have 4 tubes. It would be the same with the litmus milk. Just as we've done already, I've tried to emphasize you will probably be doing all of these labs at one time, okay, depending on which class section you're in. So you may have these tubes on top of the fermentation tubes, on top of the extracellular enzyme tubes. Okay, so you might have a large quantity of tubes. Once again, write the name of the media, write the name of the bacteria, write your group name before you ever put anything in it. I do want to emphasize that when you're writing on a tube, you can tilt the tubes, but be really careful when it's a liquid not to tilt it too far. Okay, I've seen a few spills. Luckily they were just the media. So we have the urea labeled, okay? So for the urea, that's the first one. The second one is the litmus milk. Now this particular litmus milk is a little bit dark. I think it's left over from last semester. It's almost the same color as my gloves. Your litmus milk will be a little bit lighter in blue, okay, rather than this deep color. Now with the litmus milk, there's many reactions that are going to be tested. If it uses lactose, it will produce an acidic product. If it uses the casein, it'll produce a neutral product. If you notice in the book, you'll see several different reactions. It can go from purple to pink, or it can go, starts out as a blue, okay, and it can go to a pink color. It can curd at the bottom and can turn a white color. You can have what's called peptidization, where it breaks down the proteins into the small amino acids. Then you get a clear media because everything broke up into little tiny particles. You can have an acidic product, you can have a neutral product. So with the litmus, when you go to read it next time, which will be another video on another day, you just be aware that you're going to have several different reactions that can occur. It's not going to be just one or the other like we've seen with a lot of the other videos. So as with the other one, we may have 3 tubes or 4 depending on the number of bacteria. Pretty much the procedure is going to be the same. I am not going to go over that procedure again. You would do the same aseptic technique. If this is a deep, you're going to stab it. If it's slant, you'll use the inoculating loop and spread it over the top. Same with these tubes here. They may be a screw top, they may not. They may be this form of a tube by the time you get the lab. So it's a liquid, so you're going to use the loop. If it has a screw top on the top, leave it a little bit loose. If it's one of these types of tubes, you can leave it the same. So this is for the urea and the litmus milk test.